I have to say I have a cold, so if my voice begins to go, I apologise for that. Or if I start coughing and stuttering, it is not COVID. It is just cold. On the taxi coming up, I realised I hadn't prepared the perfect talk this evening, so I did a little bit of editing. You may not have noticed it, the taxi, to shorten it a little bit. And I will do explanations that I hope will explain to everybody in the audience what it is that we're doing in my lab, and we'll talk about my lab work. But some of it might be a bit techy, a bit nerdy. I'll tell you when it gets a bit like that, and you can just sort of nod off for a minute or two, and then I'll wake you up when it gets more accessible. So I'm Paul Nurse, and I work, as you heard, on the cell cycle. And not surprisingly, given that, as I'm going to talk about work in my lab, I'm going to talk about the cell cycle. So let's start with what it is. The cell cycle is the process by which a cell reproduces itself. The cells are, of course, the basic unit of life. We all came from a single cell, so it's something you ought to be interested in once you were a cell. And it is the basis, the process of cell reproduction, of the growth and reproduction of all living things on the planet. So it's a fundamental biological process. It's called the cell cycle because it's a series of developmental events that occur from the birth of the cell to the subsequent division of that cell from 1 to 2. And the key critical ones that have to happen every cell cycle is the copying of the hereditary material, the DNA, during DNA replication, called S phase, and the subsequent separation of those duplicated molecules into the chromosomes that separate at cell reproduction, which is called mitosis for the cell divisions that go on in the majority of the cells of your body, and meiosis, where there's a specialized cell cycle for when you make gametes, that's a sperm or eggs. So this is a really central to understanding how living things work. And I spent actually all my life, and you can see from the colour of my hair, that's quite a long life. I spent all my life working on this particular topic. Specifically, how is it controlled? In other words, what regulates the temporal order of the sequence of events that occur that lead to successful reproduction? So that's what we're going to be talking about. And I'm going to explain initially the present model that is taught in textbooks. How many of you are biologists? Just put your hands up. Okay, quite a lot. So I can probably speed up a little bit. The sort of textbook explanation of how the cell cycle is controlled, which my lab did contribute to quite a lot. But actually I'm going to produce a modified version of that, which does bring a different view to it. So, I work in the Francis Crick Institute. I actually run it. And not that you ever run any research institution, that's another. This is the most exciting slide in my talk. Look at this. It's a dynamic logo. Bang, bang, bang. Okay. It goes downhill from here, all right? And this is the building, which, if you ever go to London, which I'm sure you do, you will see at the backside of St Pancras Station. And it is the biggest biological, biomedical research institution under a single roof in Europe. We have 1,500 or more research scientists in it, about nearly 2,000 staff, so it's quite a big place. So, what is the present textbook view of how the cell cycle is regulated? Well, there is a key protein. It's made up actually of two separate proteins called cyclin-dependent kinases. Again, hands up those who've heard of that. Great. That's good. Okay, we're doing well here. So, cyclin-dependent kinases, which my lab was one of the 3 labs that discovered it, is a protein kinase, which of course phosphorylates other proteins, and therefore changes, because you're introducing a fairly bulky negative charge into protein, changes how those proteins function, and so is used as a switch for many, many processes in biology. In humans, we have about 500 protein kinases, and there is a subset of them, quite a small subset, which are called cyclin-dependent kinases. There's a protein kinase component that puts on the phosphate, an activating component, which is called cyclin, which is why they're called cyclin-dependent protein kinases, or CDKs for short. Now, the way it's thought to work is shown here. There's a family of CDKs that have been identified in all eukaryotic cells. There's quite a lot of them in our human cells, rather fewer in the cells I will be talking about because they're simpler, but still a number of them. And these different cyclin-dependent kinases are thought to be different from each other in the sense that they have different substrate specificities. So they phosphorylate different substrates, which bring about the different events of the cell cycle. So there will be a CDK that occurs at the beginning of the cell cycle, G1, G1 to S phase transition when the DNA is replicated, progression through G2 and G2 into mitosis and cell division. So a family of cyclin-dependent kinases, which, although it's not always very clearly stated, are thought to appear and probably disappear at these different steps in the cell cycle to phosphorylate the proteins that are necessary for those different events, and that is the basis of the temporal order of the cell cycle. Have you had lectures that said that, more or less? Have I got it right? Yeah. Okay. Well, I'm now going to show that it's wrong, okay, as I described there, or rather, it's mostly wrong. It's not completely wrong. Now, these were discovered in the 70s and 80s initially, because I'm that old, and have been worked on ever since, and are known to control the cell cycle from the simplest eukaryote, which is yeast, which I work on, through to our cells. And many hundreds, thousands of actually people have worked on it. And there's a lot of different regulation here. I'm not going to go through it, okay? If you're interested, just cast your eyes around it. Maybe you would talk some of this stuff, okay? There is a lot of different regulation that is important. And that's led for people to study each of these different things, to study all the proteins that are involved, and then for people to do things like this. Okay? Now, I hope you can't read anything on it, can you? If you can't, blur your eyes. I don't want you to read it. I want you simply to look at it and get rather depressed. Because what happens is, they try and put all these different components together, and we have this dependent on this, and blah, 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 until it looks something like the London Underground map, only it's less clear. And this is meant to explain something. You're meant to understand something from it. And of course you don't. What insight do you have? What principles emerge from this? Apart from, my word, it does look complicated. And I got a bit tired with all of this, I have to say, over the years, because as everybody did a bit more, they added something else. And I thought it was time to think a bit more about principles that underpin the way the cell cycle is controlled. And we don't get it by just doing what we see here. And to do that, I worked on the very simple system that I work on, which is fission yeast. I'll show you a picture of it in a minute. But this is to introduce to you the cell cycle and the cyclin-dependent kinases, or some of them, that we will be talking about as we go through this talk. So this is fission yeast. It's called Schizosaccharomyces pompi, but I'll just call it yeast from now on, because it's too long a name. This is the cell cycle, which is starting here, actually, at about 7 o'clock. We have this period of called G1. Then we have S phase, that I've explained to you. period of G2, then mitosis and cell division. You can see the blue blobs in here are the nuclei that are dividing here. And what we have around here are the 4 major cyclin-dependent kinases that are found in fission yeast. There's more, as I said, in more complicated eukaryotes. There's a single protein kinase called CDK1, or CDC2. It's encoded by a gene called CDC2. And there are 4 cyclins that are thought to be responsible for the different substrate specificities as you go through. PUC1, SIG1, SIG2, and CDC13. Now, some years ago, I thought, okay, let's try and simplify this, because the principle was, to go back to this, was the following. Let's take a complicated network, let's focus on what we think is important, and then let's take away as many of those components as we can, and see if it still works. Because if we do that, we simplify the system, because biology is an informational science as well as a chemical science. We reduce the number of components we have to think about, and from that we might be able to develop principles by which it works. Okay, so it's a sort of reverse synthetic biology for those that read about these things. Synthetic biology claims to build up things from known engineering principles, and I prefer to take things apart that actually work and get the minimal thing that will actually do your job. And that way, you just focus on what matters. So, first thing I thought we could try, and the postdoc in my lab called Damien Kadrusa decided that he would try and do what I suggested, was I already explained you have a protein kinase and a cyclin. These are separate, so they've got to find each other, they've got to form stable complexes, and they've got to work together. So what we did is we simply fused the gene, so that they were a single protein. We have to put a bit of a linker in the middle. It was just a single protein, so that you removed a bit of the regulation, because you didn't now have to make the complex, you have to stabilize the complex, etc., etc. And that turned out to work. Now, this is what fission yeast looks like. It looks like a sausage, as you can see. It's a very small sausage, about 10 micrometers long. This is what wild-type cells look like. These are dividing ones with these white things in front of it. And this is wild-type. This is what we introduced into that wild-type cell, the cyclin, which actually acts at the mitosis, the last cycle it's in the cell cycle. We fused it through a linker to CDC2. And then what Damien did was to delete the endogenous CDC2 and CDC13. So they are out of the way. And these cells behave perfectly normally. They were fine with the fusion protein. Then he got excited, okay? He thought, right, I'm now going to delete the other cyclin. So he goes through the genome and deletes not only CDC13, but also SIG1, SIG2, and PUC1, and lo and behold, they're perfectly happy too. Okay, perfectly happy. So suddenly, the model I just explained doesn't look so rosy anymore, because what it means is you can drive with perfect precision all the events of the cell cycle on with one cyclin-dependent kinase. So all this story that we've invented over the previous 30 years, which had these different ones being regulated, it cannot be the whole story, because we can drive the whole thing with a single CDK. Well, encouraged by that, we actually then went and looked at a meiotic cell cycle. Remember the one I said that made gametes? Now that uses the same 4 cyclins and another 2. You don't have to worry about these, because I'm not going to talk about it anymore after this slide. So there's 6 cyclin-dependent kinases, all of which have been thought to have particular functions. We deleted those 2 too, and with a little fiddling with the expression levels, found that our fusion protein could rescue all of those too. So you only needed one cyclin-dependent kinase for both the mitotic and the meiotic cell cycle. Now, how do you explain that? I mean, this is not what is expected. Well, you explain it by a rheostat. This is shown here. This is the cell cycle, so we've got G1, S, G2, and M. And the depth of the redness, the red colour, indicates the level of cyclin-dependent kinase activity in the cell. Remember, we're driving it by a single CDK. So the notion was that it's low at the beginning of the cell cycle, that at a low level it undergoes S phase, at a higher level it inhibits S phase, at an even higher level it undergoes mitosis and cell division. And if that's true, you can explain it all by a single cyclin-dependent kinase, and you're cranking up through a rheostat to produce it. And the fact that we have so many different CDKs may be no more than that they're contributing different levels of common CDK activity throughout the whole cell cycle. And we've been misled into thinking that the major emphasis is on qualitative differences when it may be just quantitative differences. So that's quite a different way of looking at this. So when actually we first proposed it, which was in 1990, nobody, including ourselves, took it very seriously because the evidence we had then was very weak. But now the evidence we have is very strong, so I'm just going to summarise it. Now, just to re-explain this rheostat, I've rolled the cell cycle out here, so we've now got G1S, G2 through mitosis. And this is the level of CDK activity. I don't quite know the pattern, or I didn't when I drew this slide. We do now, as it increases. So I just put it up as continuous with a dotted line. Now, what this says is you need a low amount of kinase to undergo S phase, a medium amount to inhibit S phase, and a lot to undergo mitosis. Then at the end of mitosis, the activity plummets, and that's due to cyclin degradation, which is why the protein is called cyclin, which was described and discovered by, in fact in Cambridge, by my friend Tim Hunt, who described cyclin in marine invertebrates. And that's degraded to a low level, and then the whole cell cycle starts again. You know, you couldn't imagine a simpler model. Do you remember that model I explained? We're now in the land of great simplicity, which is what I prefer to be in. Now we can test this, and we can test it because we can mutate the ATP binding pocket of the protein kinase in such a way that it is made wider. This was invented by a person called Kevin Shockat, so I call it shockatizing the protein to open up the fold. And that means that a bulky ATP analog that can't be hydrolyzed can now fit into that pocket, but in theory at least, no other protein kinases in the cell. Okay? So this means using that inhibitor, we can specifically inhibit the protein kinase. Not only that, but by varying the concentration of that inhibitor, we can in vivo, in living cells, vary the amount of activity, of CDK activity you have. Basically, if you put a lot of inhibitor in there, then the activity has to be very low. If you put a small amount, then you've still got significant activity. Now, what that means is if you need a lot of activity to get mitosis, adding a small amount of inhibitor will block it. However, if you only need a little activity down here, you need to add a lot of inhibitor to block kinase. Okay? So we predict if we make such a construct, which we have, then we need only a small amount to block mitosis, a lot to block this, to block S phase. Then what happens in the middle? Now that's interesting, because if there is no arrow of time in the cell cycle, so it's irreversible. If it is in fact that the position in the cell cycle is set, as this implies, by the level of CDK activity, if we put a cell here, we can either make it go into mitosis by allowing the activity to rise, or make it go into S phase by making it reduce and then go up again, and it will go into S phase. Very simple. So that's the experiment that we did, and this is the one for going into mitosis. Okay, we had cells in G2 just before mitosis. We released them into mitosis, and this is the numbers of cells undergoing mitosis, and this is the control. You see this one going up here and then a second peak here. It's just DMSO, that's just the chemical solvent in which we dissolve the inhibitor. Then what you can see is we add increasing concentrations of that shock at inhibitor. And what I want you to point out here, you can see it, the more we add, the more we delay the peak, or it gets lower, until we get to 300 nanomolar and it doesn't go into mitosis at all. So somewhere between 100 and 300 nanomolar, we're blocking mitosis. At point, for the next slide, at 0.1 to 0.3 micromolar of the inhibitor. So you can just remember that, 0.1 to 0.3. Now, what about S phase? Will that inhibit S phase? Now, this is an experiment where we do an analysis of a population of cells and measure the DNA content in the cells to determine whether they've undergone S phase or not. So you don't have to worry about how that works, just imagine that it does work. And that allows us to determine whether the cells have not undergone DNA content or have undergone one replication or even more. Now, if we add DMSO, you can see that no cells accumulate in G1. G1 is very, very short, and we don't see any cells doing it. If we now add 1 micromolar, you'll see there's about a 30-minute delay of S phase, because you see this peak of G1 cells. As we go up in concentration, at 5 micromolar, you see here, we have a 90-minute delay. I don't have 10 on here, but we'd have a 180-minute delay. So that means to get a decent inhibition, you need up to 10 micromolar of inhibitor, compared with the 0.1 to 0.3 micromolar of needed to inhibit S phase. Got it? So that fits the hypothesis. Agree? The fun experiment really was the arrow of time experiment, which is this one here. Because now we're really manipulating cells. So what we did here is we accumulated cells in G2 with actually 1 micromolar of inhibitor. Then what we did is two things. We either washed out the inhibitor to allow the kinase to go higher, and then it should go into mitosis, which it did, or we added more inhibitor. In fact, what we did was add 10 micromolar, a pulse of it, for 30, 60 minutes, which pushed the activity down. And then we washed that out, and the cells went into S phase. And so we see that here, where there's re-replication, they go to 2C to 4C as a consequence of that. So this tells us that the whole cell cycle is being controlled in the order of events in the cell cycle simply by the level of cyclin-dependent kinase that is in those cells. None of this is understood in terms of the standard model that you have different ways of CDK activity to different substrate specificities. You explain it all with a quantity of activity, that's why I call it a quantitative model, and as a rheostat. And it couldn't be simpler. You know, if you were to design it, if it was intelligently designed, which by the way it wasn't, but if it was intelligently designed, then this is something that you would design it like this. You know, it's not got, you know, us biologists like to inhabit a very murky world of complexity. This is simple. It's a principle. This is the basis of the principle. Now, what that also suggests is that the substrates that are required to get phosphorylated, say at both S phase and mitosis, they don't appear in waves unless there's other things happening, such as enzymes called phosphatases that strip off the phosphate. But if it's just simply protein kinase rising, then what will happen is that the phosphorylation of substrates, say at S phase, doesn't go up and then go down. It will go up and bring S phase and continue to go up. Nobody had predicted that, but funnily enough, nobody had actually measured it either, which is, of course, because everybody knew it would go down, I suppose. Anyway, here we see it. This is S phase substrates. This is the beginning of the cycle. It goes up, S phase is here, and then it continues to go up, peaking to a much higher level at mitosis. Mitosis, on the other hand, mitotic substrates, don't get enough activity, because they need more enzyme, to the end of the cycle, then all the mitotic ones come up. So the basis of temporal order, just looking at these two events, but others in there, is simply that as it goes up, it initiates S phase, and later, as it's continuing to go up, it initiates mitosis. Now, I have a couple of experiments here, but I don't think I'll go into the details there, where we show that in vivo, that the kinase activity in vivo is much lower here than here. I'll just show you that for those of you who may be... right, this is when most can switch off. Okay, you stop listening if you want. What we simply did here is took cells, hit it with a shock inhibitor, killed all the turnover of the phosphates is only 2 minutes, so 20 minutes, dephosphorylates all the substrates. Then we wash out the inhibitor so that we return the protein kinase activity back in vivo. These are living cells, which is a bit like doing an in vitro biochemical assay. And then we use the mass spectrometer to measure the rate of phosphorylation. So we measure in vivo phosphorylation instead of in vitro. Now, some of you have got that, maybe not all of you, and I apologise for that. And the answer was spectacular. If we look at G1 cells, the in vivo kinase activity is down here. Mitotic cells, it's right up there, and it's in between that. And the early substrates and mid and late substrates all have different specificities. This is the first time actually that in vivo protein kinase assays have been done in a massed way. People use things like FRET sensors looking at a single one, but here we looked at 300 substrates simultaneously with this assay if you're interested in this. Okay, so here we have an explanation for all of this. This is the rheostat model. Really, CDK activity goes this way. At low level, it brings back S phase, other events later, and then mitosis, and we have these events laid out. And that's the basis of the order of the cell cycle. Now, I had a problem. This we did some years ago, but I had a problem with it because I remember what I said, but I said it very quietly and only once, was that the CDKs, there's 4 of them here, contribute to the total activity. Did you remember that sentence? I said it quietly because I wasn't sure I wanted you to pay too much attention to it, because what that predicts, if we take it seriously, is that whereas this kinase, which is the G2M kinase, the mitotic one, a complex between the CDC13 and CDC2, we should be able to get the CDK that acts at the very beginning, which is SIG2 and CDC2, to also undergo the cell cycle. And we had tried for quite a long time, and it had failed. It didn't work. Okay? So I always held back from the strong model, because I didn't like that. Okay? It failed. I won't explain how it explained, but it failed. Anyway, we decided to have another go with it, and this is with a graduate, so now we're getting up to date. So this is only been published in, actually Nature only a couple of months ago. And this was done by a graduate student, and my lab is nearly entirely graduate students. I point this out because I'm recruiting for graduate students, by the way. The next in, well, this autumn. So if anybody's interested, apply to the Crick. I'm going to take 2, I think, okay? Now, this model here, and again, this experimental practice, this is done by Saz, who came from Oxford actually, what he did is, he made this strain here, and I just will tell you what it is. CDC2, this is a temperature sensitive mutant, so this means at high temperature that protein kinase is dead, and at low temperature it works. I isolated this mutant actually in 1973, just to put it like this particular one. Okay, so that's this one here. He then put into it a CDC2, cyclin-dependent kinase, fused to a cyclin, and that was either CDC13 or SIG2. Okay, and we put it under a regulatable promoter, a tetracycline promoter, and then we had a sensor here for whether you're in mitosis. This meant that if we now shift cells up from 25, where this protein is active and so it's acting normally, to 36, we switch this off, and then the cells do not have a CDK, and we switch on the fusion protein, and we can then ask in the fusion protein at high temperature, what can the cells do? And the question is, what can they do with the SIG2 fusion or the CDC13 fusion? Got that? Yeah. All right? Nod, encourage me. Not go on. Right. Okay. So what happened was failure. Okay. That's here. This is cells going into mitosis. This is CDC13. So this is cells that are binucleated. This is CDC13 CDK1 fusion, and this is the SIG2. Nothing happened. Dead. So we confirmed what we've done several times previously by more conventional methods. But Saz didn't give up. Okay. And what he did is he looked at the various inhibitors of CDK activity that we'd identified over the years by genetic methods, and I'll say a bit about genetics at the end of my talk. I won't go through them all, but we had about 4 that were relevant there, and deleted them, because we can do that sort of thing dead easily. The argument was maybe this wasn't working because there was an inhibitor around. It had nothing to do with the problem with the catalytic activity, it had something to do with a higher level regulation. And it turned out that if he deleted a phosphatase, PP1, just one of these here, that now he got a pretty good entry into mitosis. And he made that better. And now we're going to go a bit technical again, so you can switch off if this is too much. But you've got the picture there, we've got rid of one inhibitor. Because that inhibitor, phosphatase 1, is found in a particular place in the cells, rather concentrated there. It's called the spindle pole body, which is a centrosome in eukaryotic cells. And Ian Hagen, who was my graduate student, is now in Manchester, had shown that PP1 is tethered to that spindle pole body through another protein called CUP12. And we also knew that CDK is in the same structure. And so Baz, being a clever soul, thought maybe the inhibitory effect is in the spindle pole body. And if I get rid of the tethering of the phosphatase to the spindle pole body, maybe I'll still get rescue without lots of side effects that could be happening, because PP1 phosphatase will be hitting many substrates. It's not very specific. And he had no right to work that experiment, but it did. So what he did is he used mutations that Ian Hagen had identified that tether it to the spindle pole body. And now what we see, if we look at that strain, is that the control, CDC13, is here, and the SIG2, CDC2 is here, absolutely bang on top of each other. So this means the catalytic activities of SIG2 is entirely workable, as is with CDC13. So that is the next thing that we did, that we wanted to show. And the final thing is we thought we would, the same system, the substrate specificities of these two kinases, these two fusions, again by using mass spec analysis, because what we could ask is, as we, remember here, as we bleed in the kinase here, we can now look at what substrates are phosphorylated, and when they're phosphorylated, and at what level of enzyme they're phosphorylated, and now compare the 13 fusion with the SIG2 fusion to see how they are in biochemical terms, because what I just looked at was biological, right? And this is the answer. We looked at about 250 substrates, whatever we add all those numbers up together, lots of substrate. 180 are here. This cluster is CDC13 and SIG2, it just sit right on top of each other, okay? 31 here, the SIG2 is actually higher than CDC13, even though most of these substrates are normally CDC13 substrates. The same over here with a slightly different pattern because we use an algorithm to put them together. And only 36 of the total 250 was the SIG2 lower. And even then, it wasn't, it was about 50%. And actually, we only had 11 phosphosites on about 7 proteins where it was less than 1/3. So what it means is that well over 90% of the substrates are absolutely identical. And that is, if you like, the biochemical proof that there isn't a massive qualitative difference. So our conclusion with all of this is that the principle that drives cell cycle progression is quantitative increase in CDK activity. But there are qualitative features, minor ones, which we see here. It's a bit like, Chinese Marxism. It's Chinese Marxism. It's Marxism with Chinese characteristics. This is a quantitative model of principle with minor qualitative features. And that is a very different picture from the standard model. Now I'm going to say one more thing about this, because if you think about it, this isn't so unexpected. Because given that we have a range of cyclin-dependent kinases, by the way, in our cells we have different, a collection of CDKs, CDK4, CDK6, CDK1, CDK2, etc., and a range even more of cyclins A, E, B, C, D. You probably have to learn them all off by heart at different stages. And but those must have all arisen in evolutionary time by duplication. So what that must have meant is that it was duplicated from a single gene. And that must have meant that the single gene did it all. And all we've done is resurrected something that was 1,500 million years ago at the birth of eukaryotes. And that is the principle that drives it. But when you make more complicated eukaryotes, which rely on tissues and bodies and so on, you need a more complicated regulation which imposes on the principle variations that allow you to do that. And that accounts for why, very often, when people do experiments in mammalian cells, playing around in ways a bit related to what I've just described, that you can get tissue culture cells to divide with a variety of different CDKs, but you cannot make a mouse, because it's too, you need an elaborate level of regulation to do that. But the principle is the rheostat with little additions, particularly important when we come to complicated, more complicated eukaryotes. So that's the different model. Now I'm going to skip this, which talks about other things like that, okay, and spend the last 15 minutes, something, on the problems we're now trying to solve, okay. And I've listed them here, and I just want to. So you get no answers from what happens now. It's just a potpourri of things that we're wanting to do and how we are approaching them. But I hope it's of interest to you. Now, the first thing is that the CDK activity we are looking at from the beginning to the end of the cell cycle is huge. It's going up from a couple of percent to 100%. Now, normally when people think of protein kinases, they think it's on or it's off. What this says is, no, it's a dynamic rheostat. And not only that, but the activity is varying over a very significant range. A dynamic range that could be as much as 50-fold. How is a dynamic range of 50-fold put in place? Originally, it was, of course, thought that different CDKs did it, but now we know that, in fact, you only need one CDK to do it, so that is unlikely to be the case. So what is the nature of that dynamic range? Then we have two really nice biological problems. Cells divide when they reach to a certain size. They divide because they activate the cyclin-dependent kinase activity, and to do that, they have to measure how big they are, measure their size, and input that measurement into the regulation of cyclin-dependent kinase. Now, how does the cell measure how big it is? Well, I've actually been thinking about this for nearly 50 years and not yet solved it. But I tell you, before I'm dying, I'm going to solve it. I intend to do that. Not only that, so that's an interesting problem too. Not only that, but actually the size of the cell is also set by the ploidy, that's the DNA content of the cell. So if you take a cell and double the DNA content, so you go from haploid to diploid to tetraploid, the cells double approximately in size. So the size measurement is modulated by the ploidy of the cell, the DNA content. So the cell knows how much DNA it's got. I mean, I'm not suggesting it knows it in an anthropomorphic way, but I'd say it because it makes sort of sense to everybody. The cell knows what DNA content is. It knows what size it is. It integrates these, because as I said, biology is an informational science, and we have some computing people there, so I thought I would just emphasize that. And these things are integrated together. And how the hell does the cell do it? Well, we're going to sort it, and I'm going to show you our approach to it in a moment. Then there's an issue of threshold. If you have a rheostat is a very sensitive instrument when it goes from 0 to 10%, and very insensitive when it goes from 90 to 100%. So somehow you've got to introduce thresholds into that range of rheostats. So how is that brought about? By processes of futile cycles and so on. There's different ways in which you can do it, but we are still working out how to do it. And that involves dynamic regulatory behaviour. Now, how do you approach problems like this? Well, just like the way, in fact, the work I've just described, it was underpinned by a lot of genetics, which I didn't describe to you, that was done over the years. We are using genetic approaches to identify the components, the molecules that could have some role in this, and then testing whether they do. And this is work being done by Jackie Hales, who just retired, having worked with me for 35 years, and was my first graduate student, in actual fact. And she uses, the approach she used was what's called haploinsufficiency. And I want to explain why this is so important. If you've got a regulatory network, and we've got a network going on here, one way of identifying whether something is likely to be regulatory is to reduce or increase the activity of a component a relatively small amount and see whether that has any impact on the biological phenotype that you're looking at. Because most experiments are much cruder than that. They'll, for example, hit the gene with a sledgehammer, knock it down from 100% to 2%, and make almost anything in the cell rate limiting. But if something is really rate limiting and regulatory, it'll be responsive to just small changes. Now, it's a lot of work to do small changes, but one simple way is to take a diploid and kill one of the genes, because it have two genes, which will reduce most of the time activity from 100% to 50%. And so you can then look to see, do they have any impact on the phenotype that you're interested in? And in this case, it was the size at which you underwent mitosis, because that's the first problem, okay? So that's described here, but I don't need to go through it. Did haploinsufficiency and asked what genes either increase or decrease the size of mitosis when you change it by a relatively small amount. And actually, oh yes, and I didn't tell you, to do this experiment, we had to do something else first. Fission yeast has 5,000 genes. And we led a consortium that deleted every single gene, all 5,000 genes. That was actually led by Jackie. It drove her completely mad. She says it hastened her retirement and demise in life. 5,000 genes, all deleted, all arrayed out, so that we can now look at the impact on the biological phenotype. In this case, just the length of the cell at which it divides, a very subtle one. To ask if you reduce the level of activity to half, what happens. And this is what came out of that blind screen of all the genes. The first was a whole set of genes that we'd already identified as being important. And you might say, well, it's boring, but it just shows you what you were doing was right. So look what we got here. We got CDC2, okay, CDC13 that you heard about, which acts at mitosis, a gene which Jackie discovered in her PhD and was still working on to work out what it does. Genes which some of you who may have heard of this in your classes, WE1 and CDC25, POM1, another gene that my lab showed worked in the mitotic cycle. And these are all co-regulators of the G2 mitosis transition. So we're in the right place. somewhere in here is how the cell is measuring its size and ploiding. Don't know yet how it does it, but that's some way of doing it. Then we have a whole network of other genes over here that we have not a clue about. All of these are part of the nuclear pore that is important for importing proteins into the nucleus and exporting. And so nuclear import and export plays an important role, one that we didn't know about, but we now do. And then we had another couple of things here which I don't understand and we don't need to talk about. So that was one approach. This is standard genetics, okay? Huge amount of work. Deleting 5,000 genes took 7 years, okay? 7 years. And a South Korean consortium worked with us. Then screening for these sorts of mutants. And then validating. A huge amount of work. Then another, this time a postdoc in my lab, used a different approach, which is just looking for haploid mutants from the 5,000 that have been deleted. It's only 3,000 that were viable, or 3,500. And look for cells that were advanced into mitosis. This is what wild type looks like. This is the mutants. I found the first one in Edinburgh, I called it Wee1. I thought it's a funny name. Not so sure now, so many years later. But you see, these are really advanced into mitosis before they've grown enough. So that was the lock that, that was unlocked to do this. And then we've got all these mutants here, which were what I call semi-Wee and are involved in a variety of things. These are involved in the G2M transition. You'll see some genes, Wee1 and so on, and the phosphatases here, nutritional sensing, and other things that we don't really know how they work. Other candidates. Put all of these together, and we've got around, and adding a few more that I haven't told you about, we've got around 40 genes that are in the right territory. So then what we decided to do, and this is why this is so preliminary, we decided to measure the level of all of these proteins as they proceed through the cycle, just to make the hypothesis, maybe the level of these proteins has something to do with measuring cell size, and so they would change as the size gets bigger. And we did that, and this is done by Scott Curran. We did it by a method, fission yeast, as it goes through the cell cycle, gets longer, so you know where it is in the cell cycle just by measuring it, okay? Which is the reason why we work on it. And you can put it through a fancy cell sorter, which automatically measures the length and also measures, if we make the proteins coloured by tagging them with another a chromophore, then it will make the, we can measure the level of protein. And we get fantastic data. Look at this. This is the level of CDC2 as you go through the cell cycle. I mean, isn't it beautiful? This is, I mean, you don't see data. Us physicists get this stuff all the time, but we get data. And this is great. This one was one that does this beautiful change, CDC25. This is the phosphatase that regulates CDC2. I put all this on a heat map. Amazingly, all these are the regulators, and the only two that underwent significant change in the total cell was CDC13, which we knew anyway because it's cycling, and CDC25. Now you could say this is a lot of work for not discovering something. What it meant is we don't have to think about all these other ones. Okay, we are confining our attention to relatively few genes. If I looked at the G1S transition, this is the G2 to mitosis where cell size is sensitive, the periodicity was much more common. I don't want to talk about that particularly, but or at all in fact, but it's much more common. Now, I want to finish my talk by just talking about 3 proteins that turn out to be quite important now, all core of the cyclin-dependent kinases. Now, CDC13, this is total cell. You see what happens? It goes up as cells increase in mass, then it's degraded as they go through mitosis and cell division down to here. Okay. Now, what was interesting was we could also, using this time a light microscopic method. Oh, by the way, that image stream, we look at 100,000 cells. That's why the data is so good. But under the microscope, we can look at about 10,000 and use computational methods. So it's still pretty good. Or between 5 and 10,000. To be more precise. And what we found is that the rate of change within the nucleus was even greater than in the cell. We looked at the nucleus because of what I told you before, that we thought nuclear transport was important. So that was CDC13. CDC25 is a phosphatase that activates CDC2 protein kinase. And that is because WEE1 is an inhibitory kinase that puts a phosphate in the ATP binding site again, and CDC25 is the phosphatase that takes it off. And what we found there was that actually it falls through the first half of the cycle and then increases during the second half of the cycle, unexpected. If we now looked at amount rather than concentration, actually it's flat and then just goes up in the second half of the cycle, or second third of the cycle. We see also a more pronounced change within the nucleus. CDC2 is interesting because CDC2 is constant in concentration in the cell, but in the nucleus it increases. So this tells us that the size, maybe it's measuring something in the nucleus, which is dependent upon the cell itself and the cell size itself. I don't know, and this is just what we're doing at the moment. Now I'm going to give you some thinking about this, and then I'm close to stopping. Yeah, I'll dump the end of the talk so I can stop. Okay. Now I put up here CDC13 and CDC25. Now CDC13 could have a role in size sensing, okay, because it just accumulates as the cell goes up. It can't be just that, so it's got to be other things as well, but this could be a major role of it. But then what's interesting is that this is the rheostat problem, because if it is, then this is fine measuring size, but this is, you're going up from 70, 80% to 90, 100%. And you haven't got a good threshold transition, It's not a good threshold transition. But that's rescued because WEE1 and CDC25, between them, produce a futile cycle. Now I haven't got that up here, but basically WEE1 is inhibitory, CDC25 is activating. So you get an equilibrium. And as you go through the cycle, as you showed, I can use this one because I've got it here. As you go through the cycle, CDC25 is reducing, which means the futile cycle isn't operative because it's not taking it on. But as it starts to increase, it will now move the cell into a futile cycle mode. And that has the potential now to introduce a dynamic instability that will lead to a switch behavior because you're amplifying. So what we're thinking may be happening is this is, say, CDK activity, which goes up gradually, and then when you switch into the futile cycle, you become bistable, which allows now the potential to have a sudden jump in activity, so you have an irreversible entry into mitosis. And indeed, what we've shown is if we eliminate that, you get many mitotic errors because it can't do it properly. So what we're thinking about, and this is still speculative, is that there is, if you like, a sort of deterministic size sensing role, which gets you to a state where you now are approximately the right size and you are big enough to divide. Then you switch on the futile cycle, which will activate at a more variable time, but will mean you can go into mitosis very efficiently, as you see here. So we're working on it. Might be wrong. I have a feeling it's, but it might be. Okay? Now, I was going to end with in vivo CDK activities done by another graduate student in my lab who came from Cambridge, this one, and he did that bit, and now we've done more on it, and that's there. And I'm going to finish here. It's back to and just do a quick summary. Okay? So what have I said? The qualitative model is the one that you will read in the textbooks. A range of different CDKs activated at different times that bring about temporal order. I do not think that is the principle by which the cell cycle is ordered. I think it's a quantitative model where you look at total CDK activity. However, I do think that in more complicated eukaryotes, such as our cells, qualitative features may be needed for dealing with development and multicellularity and the more complex things our cells do. But the quantitative feature is the one that actually drives it. I think that the eukaryotic system of a cyclin-dependent kinase was invented, if you like, at the birth of eukaryotic cells 1,500 million years ago. Oh, by the way, I'm doing a really rather bizarre experiment at the moment. We're using bioinformatics to try and predict what the CDK looked like 1,500 million years ago, which is a long time. And we've just made it in the lab, and I want to see how it performs today. Now, this is just a joke experiment. We don't know if it's the real one, but it's the best approximation, just to see, what are its properties. Can it measure size and choroidity? If it does, maybe that's what it was doing then. Who knows? We'll see. There may be several versions of it, and we'll test them all if necessary. So that's the second thing. The third one is, there are these problems. How does a cell measure its size? How does it measure its choroidity? How does it get dynamic stuff? We have all the components that we have to think about. We just have to now work our way through it. You want to do a PhD in my lab? You could actually doing that. So with that, we have, of course, the last slide. Well, it didn't happen. Let's try again. Go away. Let's try it again. Ah! There. Fine, that's the end of my